Hello, this is Professor Torsen. This is the recorded lecture for Chapter 15. Chapter 15 presents various DNA technologies. We are going to begin our discussion by looking at GMOs, or genetically modified organisms. I'll let you read through the information on slide 3 on your own. GMOs, or genetically modified organisms, have been around for centuries. Humans have selectively bred organisms, including crops and domesticated animals, and anytime humans selectively bred or crossed different crops, that was a form of genetic modification. The difference is that now we have the technologies and the capabilities to modify the genetics of an organism more precisely in the lab. Genetic modification can be used to increase the yield of crops, decrease the need for pesticides, or increase the nutritional value. For example, bananas, shown here on slide 4, have very large seeds and very little fruit. However, this does not resemble the bananas that we see in the grocery store. Another example is golden rice. In certain areas of the world, vitamin A deficiency is a problem. Individuals who don't have enough vitamin A will have eye issues. Vitamin A is needed for development of the eyes. So vitamin A deficiency can cause blindness. Golden rice, pictured here on slide 4, is rice that has had the beta-carotene gene inserted. Beta-carotene is the precursor to vitamin A, and so individuals who have access to this golden rice have an additional source of beta-carotene and therefore a different source of vitamin A. There are a few glossary terms when discussing different DNA technologies. You can see these on slide 5. PCR, or polymerase chain reaction, is a technique used in lab to target particular regions of DNA and then amplify that or create many, many copies. PCR consists of 3 stages: the denaturing stage, the annealing stage, and the extending stage. Here on slide 6, we see we are starting with a piece of DNA. DNA we know is double-stranded, which are these 2 blue lines, and not represented here are the hydrogen bonds that are holding these 2 strands of DNA together. The first stage is the denaturing stage. The sample is heated, and when the sample is heated, these 2 strands of DNA are separated. Heating the sample breaks the hydrogen bonds that are holding the 2 strands of DNA together. In the second stage, called the annealing stage, a primer is added. And a primer is a specific sequence of nucleotides, and it is targeted to the specific genes of interest. The last stage is the extending stage. DNA polymerase, which is the enzyme that we have seen when we were looking at DNA replication, DNA polymerase is able to attach to those primers and then continue synthesizing the complementary strand. We see this here in this figure. So here's the primer in this darker blue, and in this lighter blue, we see the new strand of DNA that has been synthesized by the enzyme DNA polymerase. So after the end of this first PCR cycle, we have 2 copies of DNA when we started with 1. So this PCR cycle can be repeated many times to quickly amplify or create a large number of copies of DNA. This you can see on slide 7. We start with 1 copy of DNA, and at the end of the first cycle, we have 2. Then we can repeat the denaturing, annealing, and extending stages and create 4 copies. You can see that every time we go through another cycle of PCR, we double the amount of DNA. So this can be used to create a large number of copies, and this we call amplification. Gel electrophoresis is another technique used to look at DNA. Some people call it DNA fingerprinting because it will separate mixtures of DNA fragments. It separates mixtures of DNA fragments based on size. Here on slide 8, you see the typical setup of gel electrophoresis. Here in this chamber, we have the gel, and the red and the black wires are the wires connected to the power source. Here we can see an image of the gel itself, and at the top there are these indentations. These indentations are called wells, and the samples are placed into the wells. You can see that the power source is then connected with the negative electrode closest to the wells and the positive electrode further away from the wells. Because DNA is negatively charged, having the negative electrode up top and the positive electrode towards the bottom means DNA will travel through the gel from the wells towards the opposite side. The gel slows down larger molecules. So larger molecules or larger fragments of DNA don't move as far. Here you can see an example of a completed gel. So these blue lines here are called bands, and each band represents a certain sized fragment. The bands that are down here towards the bottom of the gel that have moved the furthest away from the well, these are shorter or smaller fragments. The bigger fragments, again, get slowed down by the gel and don't move as quickly or as far. Samples are usually compared to a standard or marker that creates a bit of a guideline in order to estimate the size of these fragments. That concludes the recorded lecture for Chapter 15. Thanks for watching.